Welcome to my presentation on fine-scale interannual distributions of meso-zooplankton in the Northern California current. The objectives of this project were to analyze the fine-scale plankton distributions on the Newport hydrographic line sampled in the summers of 2016, 2018, and 2019. This is a historical transect on the Oregon coast that has been sampled for many years. We also wanted to investigate the environmental drivers of these plankton distributions. We did this by a combination of underwater imaging and machine learning, which you will see is a powerful approach. On the left-hand side here, we have the PICES imaging system, which has different cameras and environmental sensors, such as one for chlorophyll A and dissolved oxygen. It is towed behind the ship in an undulating fashion. On the right-hand side, here we have example images of plankton that PICES imaged, and we sample everything from large phytoplankton through the crustaceans, including our important copepods, gelatinous plankton, such as ctenophores and siphonophores, and then we image also the larval fish, which is where PICES gets its name from, for instance here myctophid or lanternfish larvae. A short note on how we analyze the plankton images. We image about 10 to 50 million plankton and particles per hour, depending on the productivity of the system. This can lead to a total of up to 1 billion for a cruise. The images are classified into their taxonomic groups using a convolutional neural net. And in this flowchart, I show you a high-level overview of our processing pipeline. When we get off the ship with the hard drives and videos, we upload these to Oregon State's computing center, and from there we control a pipeline that gets the videos up on NSF's Exceed supercomputing platform. Here we do the actual segmentation and classification of the plankton images. An important step afterwards is the post-processing, which includes a lot of confusion matrices to make sure the classification is performing well. I won't go into more details here, but please ask questions later if you want. We recently open sourced our pipeline on Zenodo. That publication includes everything to set up an image processing pipeline, includes example data, code, and extensive documentation. I invite you to check it out. This map shows the study area on the Oregon Shelf. The triangles show the Newport hydrographic line stations where NOAA is historically sampling for plankton and water. Beneath, the solid black line is the PICES transect we did in 16, 18, and 19. On the right hand, we see the PICES towpath overlaid on bathymetry. We start in shallow water of circa 50 meters, and when we get into deeper water, we start undulating to our max depth of 100 meters. The spatial resolution of the data we gather is in the range of centimeters, which is then binned into 1 meter vertical intervals for analyses. And when we look at distribution maps, then we also use creek maps, but these are not used for the actual analyses. To start off, we used NMDS analysis for data exploration. Based on that, we then further investigated spatial patterns using random forests, which is a tree-based machine learning approach. Random forests is an ensemble learning method that combines many decision trees that are grown based on random subsets of the data and predictors. We used an unsupervised random forest classification and PAM clustering to find spatial clusters of species distributions. We then used the same taxa distribution to predict the clusters so that we could take advantage of random forest's excellent way of showing which predictors are driving a model. We then used the environmental data to predict the clusters. Environment data was not used to make the clustering before, so it is interesting to see how the environment agrees with the clusters. Let's jump into the results, and I'll start off with the environmental setting. For this figure and the next ones, we have depth on the y-axis going down to our max depth of 100 meters, and on the x-axis we go from 0 kilometers on the transect to our maximum distance on the left. What we can see is that water temperature in 2016 was quite a bit lower, while in 18 and 19 we got temperatures of over 17.5 degrees Celsius in surface waters. Here we look at dissolved oxygen, and the takeaway is that in 2018 we found lower oxygen waters at the surface, but also at depth compared to 2016 and 19. You can see there's a thick layer of low oxygen water on the shelf here. Now let's look at chlorophyll A. Note that I had to plot this in log x+1 scale because of high interannual differences. In 2016, we found a subsurface layer of high chlorophyll A reaching up to 30 micrograms per liter. In 2018, on the other hand, chlorophyll A was pretty low. 2019 then showed not much structure, but a higher background concentration as shown by the light green color. Now let's look at distributions of different taxa. The figures are now showing concentrations of taxa in individuals per cubic meter. Pteropods were distributed on shelf and deep in 2016, while in 2018 and 19 distributions tended to be more offshore. Now we're looking at the important and abundant but rather small copepod Orthona. These are important prey for many other crustaceans and ichthyoplankton. Orthona was distributed rather similar in 16, 18 and 19. With some individuals in deeper water on the shelf, but the bulk being distributed shallower. In 2018, distributions were concentrated more offshore, and 18 and 19 generally had substantially higher concentrations than 2016. As a higher trophic level example, let's look at mactophyid or lanternfish. These were distributed quite patchy, but with a clear tendency to off-shelf distribution. Looking at these distributions, we can see how underwater imaging enables us to see this kind of detail, which would likely be very difficult to sample with nets. Now we'll talk about our analyses, and we start with the non-metric multidimensional scaling, or more commonly known as NMDS plot. The black dots show our species locations, while the blue arrows depict the environmental vectors for temperature, oxygen, chlorophyll, and density. Note that I do not show the site points, but rather just plotted the locations of different site combinations by using ellipses. The blue ellipse shows stations that are shallow and off-shelf. The purple ellipse shows shallow and on-shelf locations. The red ellipse shows deep and off-shelf locations, while the green ellipse shows deep and on-shelf locations. We can see that the sites separated out by horizontal and vertical strata quite a bit. Remember that in the NMDS, these locations are drawn solely on the dimensionality reduction of our 60 plus taxa and their concentrations. This is in contrast to the 2018 NMDS, where everything is more clumped and not separating out as well. Species locations are more bunched and environmental vectors are much shorter, and the shelf depth strata ellipses don't separate out at all. In 2019, everything expanded out more again. The environmental signal was stronger, and the ellipses show that locations separated out by off-shelf versus on-shelf. We took this as a jumping-off point for our further analyses. To tease apart the similarities and differences between the 60-plus taxa distributions between the years and investigate how the distributions were driven by the environment, we clustered the taxa distributions. We used partitioning around meadoids, clustering, short PAM, on a random forest distance matrix to find the optimal number of distribution clusters per year. The analysis found that there were 5 clusters in 2016, 2 clusters in 2018, and 5 clusters again in 2019. Comparing the spatial locations of the clusters, here, we found that the 5 clusters in 2016 were clear and distinct. Remember that these clusters did not include the environmental signal, but only species concentrations. In 2018, the clusters, only 2, were not very distinct at all, a striking difference to 2016. 2019, on the other hand, showed something akin to 2016 again, however, not as distinct and not as continuous. We can see, for instance, cluster 5 here in purple, that we found shallow mid-transect here, as well as deep off-shelf here. The supervised classification of the clusters using the taxa concentrations only showed us who was driving the clusters, strictly statistically speaking. Here I color-coded the top 10 taxa for cluster 1. So the reappearance of the green phytoplankton chains here, here, and here shows that phytoplankton was an important predictor in most clusters. However, we see that clusters 2 and 4 don't share much color coding with the other clusters and were quite different from clusters 1, 3, and 5. For 2018, I only color-coded the top 3 to show the similarity between the clusters. This hints at the more homogeneous distributions we also saw in the NMDS plot earlier and in the cluster locations on the last slide. For 2019, I color-coded an interesting group of organisms that we didn't pay much attention to yet. Many protists appeared in the top 10 of each clusters. Let's look at the clusters as a function of the environment. What we see here are similar to the last slide, the importances of the environmental variables in predicting the different clusters. On the y-axis, we have the variables sorted generally from top to bottom according to their importance, and the x-axis shows that importance scaled to 100 for better comparison. The bars are color-coded according to their cluster. In 2016, we were able to predict the clusters with an accuracy of over 85%, which is quite good given that we determined the clusters only on the species concentrations. So the distance along the transect was for all 5 clusters of utmost importance. This is very interesting as distance along the transect contains the on-shelf off-shelf signal. We further see that while salinity was the most important predictor for cluster 1 in red, for other clusters this was different. What is standing out most is probably cluster 3, where depth and oxygen are of the highest importance after distance. Interestingly, cluster 3 was the cluster in 2016 that was so to say filling in the gaps, not one of the large distinct ones. In 2018, the error rate was only 9%, but somewhat expected as there were now only 2 clusters. The relative importance of distance along the transect was now even higher compared to the other variables. Noteworthy is that the second most important predictor, on average, was oxygen. In 2018, oxygen in surface waters and close to the shelf was substantially lower compared to 2016 and 19, likely having an effect on species distributions. In 2019, we saw a similar high effect of distance along the transect, while the second most important variable was chlorophyll. In 2019, we found a substantially higher background concentration of chlorophyll A compared to 16 and 18, which was likely driving to an extent distributions of key taxa. The higher relative importance of environmental variables, other than distance along the transect in 2016, matches the NMDS environmental vectors that we saw were strongest in 2016 and weaker in 18 and 19. In summary, underwater imaging and AI can give us an unprecedented look into the fine-scale distributions of plankton. We found striking interannual differences in temperature, oxygen, and chlorophyll A. We found a clear clustering of taxon-specific spatial distributions in 2016, a weak clustering in 2018, and an intermediary clustering in 2019. In 2019, we also found a high importance of protists in the clusters in combination with higher temperatures. This could indicate a higher activity of the microbial food web. Distance along the transect was important for predicting clusters. This is interesting because it is likely a proxy for other variables that vary with distance to shore, for instance offshore transport and the upwelling front. Important predictors aligned with the primary environmental conditions of the 3 years, in 2018, O2, and in 2019, chlorophyll A. Next, we will incorporate the upwelling front and the distance to it as explanatory variables, and we will work on a multi-year model with indicators such as the PDO and the QT. We thank the different people and institutions listed on this slide for their help over the years. Here are my references. And with that, I thank you for listening. Please let me know if you have any questions.